Filters in PMart can be categorized into two distinct groups, filtering out molecules or filtering out samples. All filters come with plots to visualize how much data are removed with each selection of a parameter value, and options below each plot for modifying the plot like changing labels, colors, etc. The first of the molecule filters, simply called the Biomolecule Filter, removes all locally observed molecules and can be used with any data type. The next molecule filters are for mass spectrometry and NMR-based data types. For transcript-only filters, skip to the end of the video. There is an imd-ANOVA filter for removing biomolecules without enough samples for downstream statistical comparisons, since both the downstream G-test and the ANOVA test require a minimum number of samples. We also have a coefficient of variation filter, which removes biomolecules with highly unusual variability, though this filter is not often applied. For peptide data, there is a molecule filter to remove lowly supported proteins which have only a few peptides supporting the existence of the protein in the sample. This filter also includes an option to remove degenerate or redundant peptides, which are cases where peptides map to several different proteins. There is also a capability to include custom filters for any Omics dataset. This can remove molecules or samples. For example, if your proteomics identification software does not automatically remove contaminants, you can search for key patterns in biomolecule identifiers to remove these. Now, in terms of sample filters, the robust Mahalanobis distance, or RMD filter, can be used to identify outlier samples to filter based on the distributional properties of each sample's abundance. In this plot, samples above the threshold may be unreliable or biased results for downstream statistics. Care should be taken with the selections here, as most omics experiments have a precious number of samples. Here we have an example of a sample outlier that should be filtered out. Once you are finished selecting filters, click Review and Apply All Filters to get a summary of filter selections. If you are happy with these selections, click Apply and move on to the next tab if appropriate. In terms of the transcriptomics filters, we have a total count filter which removes transcripts with less than some total count across all samples. We also have RNA-seq filters that can remove samples based on either a minimum specified library size or a minimum number of non-zero gene counts. The RNA-seq filters are provided in case there are any samples that exhibit egregious behavior that have not already been removed. These filters are not designed for the quality control of the identification portion of the bioinformatics pipeline. If you are interested in RNA-seq alignment and sequence quality, you can use tools like FastQC. Helpful graphics will show how much data remain after filter selections. Once completed, click Review and Apply Filters.